First of all, thank you very much to Pierre and Cindy, especially for giving me the chance to be here. I think it's a real privilege, and I've really enjoyed these 2 remarkable days. I'll remember them. Natalie was telling us yesterday that we should add a P. We should add a P to the promise and the power. So I've done that. At least, there it is. I've added the product she was encouraging us, all of us, to try to add to our talks. In fact, I think that in the field of livestock genomics, and especially dairy cattle genetics, genomics has given results beyond expectation. And I think that this has not been publicized enough outside of the world of livestock genetics. And I thought what I will try to do today is to make sure that the audience here at least is aware of the quite remarkable achievements, I think, that have been realized in the field in which I'm active. It's not at all my work, certainly not my work only. I feel that I'm representing a community here. And so more than talking about my work, there will be one slide, or the work of my lab. It will be the work of many, many people, which I think have generated really interesting results. Oh, there we go. So you will see the titles go from empirical to biometrical to marker-assisted selection and then genomic selection. So I'm essentially going to give you a history starting very early on. So Svante Pääbo told us that one of the things that is making us different from Neanderthal is our capacity of technological innovation. And I think that one can say that one of these genius moments that humans have had is to domesticate plants and animals, something that happened approximately 10,000 years ago. Like you all know, we have good reasons to believe that this is one of the things that really changed our social fabric. So there is a societal metamorphosis, if you want, that results from this initiative, if I may say so. But for the animals as well that we domesticated, this has had drastic phenotypic consequences. This is a painting here that dates back 2,500 years ago, where you see herders or farmers with cows. And if you look carefully at this thing, you will see that these animals are very different from the wild-type auroch for which they were derived, let's say, another 2,000 years before. So you see major changes in their morphology. And if you had been there, you would have been size differences, behavioral differences. So in fact, man has not waited for transgenic techniques to really engineer the genome of the domestic plants and animals that he lived with. I think it's particularly striking when you look at dogs and, for instance, the pigeons that Darwin liked so much. If you try to quantify the phenotypic variation between these animals that for whom the latest common ancestor is quite often a few hundred years in the past, you see more diversity than what you would see between typical wild-type species that have diverged several million years before. And maybe along those lines, just mentioning one thing related to uncovering the function of the elements in our genome, especially the genes. We've talked yesterday and heard yesterday and today about the need to be able to dig into the function. And there are a variety of ways of doing that, but one way is to close the so-called phenotype gap. So there are major efforts in mouse genetics to try to have phenotype-driven mutagenesis screens. And I would argue that we should actually take advantage of one of the largest phenotype-driven screen that has ever been conducted by people that have domesticated throughout husbandry. And to give you an example of that, a recent Nature paper coming from Dave Anderson's lab identifies a gene that allows horses to dance, a phenotype that we would only see if we live with these horses that we love. It would be impossible to identify a phenotype like that with the mouse, I believe. And I think there are many other very interesting phenotypes that can be exploited in these domestic animals. So for thousands of years, people empirically molded domestic animals in the direction in which they consider to be favorable, just by empirical selection, by mass selection, identifying the animals that looked the most along the desires of the people as the parents of the next generation. And this has generated the kinds of images that I showed in the previous slides. Things didn't change until approximately the beginning of the 20th century, which I would call the beginning of biometrical selection, when genetics was discovered. Things started pretty badly in a tension between Mendelism and Galtonism. So we had rediscovered the laws of Mendel, but a number of people, including this guy here, Galton, pretended that the laws of heredity, as stipulated by Mendel, only apply to exceptional phenotypes, but that for the majority of phenotypes, including those that are of interest in livestock breeding and plant breeding, that it was probably something else that was happening, another mechanism, so-called blending inheritance. And it took geniuses a number of years, and people like R.A. Fisher and Sewell Wright, whose pictures are shown here, to at some point propose a theory that would reconcile these divergent views. Essentially, what R.A. Fisher proposed is something that is now referred to as the infinitesimal model. That is that the variation that one observed for the quantitative traits that constitute the majority of medically and agronomically important traits, so the differences are due to a number of environmental effects, but also genes that are Mendelian in their mode of action, but there are many of them, so that the distribution in the population is explained by the fact that there are a large number of them. And in the infinitesimal model, this large number is very large, and the effect of individual genes is extremely small. Some people consider that this proposal was primarily driven by the fact that it was mathematically tractable, but few people a number of years ago would have considered that this was in fact reflecting the biology. Now, so if you work what animal breeders and plant breeders do using this model, you define something which is called the breeding value of the individual, which is in fact just the summation of the allelic effect at this large number of genes. And because the constituent genes behave like Mendelian factors, one can predict how they disseminate in the population. One can say that offspring share 50% of their genes with each one of their parents, 25% on average with each one of their grandparents. And so you can predict the correlations that are expected between the breeding values and therefore the phenotypes of animals whose genealogical connections are known. And people like, especially Henderson, have expanded on this theory using mixed model methodology, and more specifically something that is called the individual animal model, to essentially develop a sophisticated method that allows you to ideally exploit known genealogies within your population together with so-called fixed environmental effects to get the best linear, unbiased predictors of the breeding value of each animal. And this certainly has played a major role in the advances that we have witnessed over the last 50 or 60 years in terms of increase in genetic response and phenotypic performance. Now, this quantitative genetics theory on its own was insufficient. It's combined with so-called design of breeding programs in which people try to maximize accuracy of selection, intensity of selection, minimize generation interval to get this optimal genetic response. And I'll just show you one example of an outcome like that, which is right in dairy cattle genetics, where I want to invite you in. So what is done, the secret of the genetic progress in dairy cattle is the identification of elite sires, which by means of artificial insemination can have sometimes millions of dollars. There are a number of companies around the world that are manufacturing these elite sires. How do they do that? They use Henderson's equation to identify so-called bull sires and bull dams. So this is based on estimating the breeding value of everybody in the population based on known genealogy and identifying the individuals that have the top breeding value. Very often, embryo transfer is used to have a crop of individuals, of which half will be males and potentially the future champion. Now, it turns out that in the economic context that applies today, the value of these animals, which is predicted to be the average breeding value of the parents, is not an attractive commercial proposition. You have within these guys that are all super candidates, you have to identify the best ones. And this is done by a process called progeny testing, which requires to generate approximately $100 for each one of these candidates. In countries, the largest countries, people would produce several hundreds of these candidates every year. You wait until these daughters are producing milk, which requires them to become puber, to be inseminated, to have a calf, and only then do they start to produce milk. And essentially what you try to do is you try to guess how good a cow this guy would have been had it been a female. You essentially try to estimate how good his breeding value is based on the performances of his daughters. Now, when you have done that for all these crop of good candidates, you can identify the one which is the best, and this is becoming a good commercial proposition. The whole process takes 5 to 6 years and costs to generate one of these guys around $350,000. And so the industry has functioned along this principle for a very long time, and very effectively. So this shows you the evolution in terms of average milk production per cow in the United States between 1960 and 2000. It has increased quite dramatically, and the two curves here show the highest one, the phenotype, and the other one, the part of the progress that is due to genetics. So the vast majority of the progress that we have witnessed over the last X many years is in fact genetic in nature. And this without having any knowledge about the genes that were manipulated. By the way, more fundamentally, one can ask the question about the architecture, the genetic architecture that allows this adaptability, that allows this response which doesn't seem to have an end. So the variation is not, there's still as much variation now as 60 years ago. And essentially, we can move this population in whatever direction we want. There seems to be enough variation hidden in the population or generated at each generation so that we can actually move it wherever we want. And in fact, nobody knows exactly what the secret is in terms of molecular architecture of this adaptability of domestic animals, but certainly reflecting what is happening in the wild as well. So for a number of years, things were going very well using this biometrical selection approach. And then, out of Canada came the cystic fibrosis gene, and then similarly the Huntington's disease gene, and it generated within the animal genetics community this paradigm that one might do the same thing with our animals and therefore potentially replace this progeny testing scheme, which takes 5 years and $350,000, by a screen of the DNA so that one might maybe at birth or even before identify amongst the candidates which would be the best one in terms of breeding value and directly commercialize it as an elite bull. And I learned some stuff yesterday about many things yesterday, but including the hype cycle. And essentially, so I participated in this hype. And so at that time, we were, without any doubt, at the tip here of the peak of inflated expectations. And I call that the period of micro-assisted selection. Many people like me wasted a lot of their time doing these kinds of exercises with, like, as Eric Lander was saying yesterday, it's not just today assigned to a student. It has to be a pretty good student still, but it has indeed changed quite a bit. So essentially what we would do is we would go after the genes that would explain why some animals are better producers than other ones. And typically this was a 3-stage process. We would map what we call QTL, and this was based on linkage analysis, fine map QTL based on linkage disequilibrium after having generated a denser marker map in the regions of interest. And then we would identify the causative genes and variants. It's actually quite interesting to see that the last step, people seem to say it's obvious to do that. It's absolutely not obvious to do that. And I think there is still a lot of discussion about what the right methodologies are to do that, whether it's in animal genetics, human genetics, or plant genetics. So after many years, people, certainly you can go in databases. There are more than 1,000 QTLs in animals reported. There are, in fact, very few that have been fine mapped for a number of reasons. I say 100 or less. I think it's much less than 100. And if you say for how many have we gone the entire way, it's certainly less than 10. This is the slide about the number of, so my lab has been playing with these kinds of things for a while, and I think that we have uncovered a number of interesting observation, interesting biology. Just a few examples. And I'm coming from Belgium, so this is a Belgian blue animal. It's the equivalent of Jean-Claude Van Damme due to a mutation in myostatin. And then, we discover other things, microRNAs, and more recently, pieces of DNA that move around via circular intermediates. But the problem is that when we show these results to our founders, which are usually companies, well, they were essentially not happy. They said, it's nice, these examples, but if I look at practical applications, they considered that it was insufficient. And we suddenly, despite our nice papers, jumped into the hole of the trough of disillusionment. And miracles happen, and in our case, there were two miracles happening approximately at the same time. So the first one was a paper by famous people here, Meusen and Goddard. They proposed an approach which was radically different from what had been done before. Essentially, to summarize the key ideas, first of all, it was doing GWAS, but fitting all the markers simultaneously. So if you go back to Eric Lander's genetics paper about QTL mapping, there is a paragraph on simultaneous search. Essentially, this method does that. But maybe most importantly, it was going against some sort of paranoia you saw on one of the previous slides, where in human genetics, there is a real fear of publicizing false positives, and this is for good reasons. But so what these people said, let's forget about these very stringent thresholds and let's allow ourselves to go and look at markers which are not significant sensu stricto, but where there might be a signal. And they developed methods to try to go and get this signal, the purpose not being to identify the loci that contribute to the variation, but rather being very accurate in predicting the breeding value. And essentially, their simulations told us that maybe this might work and generate estimates of breeding values which would be better than progeny testing in some circumstances. Now, it was very nice to say that it was a very nice theoretical proposition, but it required markers which at the time were not available. Fortunately, sequencing technology being what it is, things went very fast. And also for the bovine, a draft sequence was generated. Tom Kasky mentioned it yesterday, and the role that Canada played into that, together with a large number of SNPs. And so not only did we have the sequence, but the private sector generated these fantastic tools for interrogating SNPs. And very quickly, a 50K, which is not a lot, a 50K SNP became available for the bovine. So it was it was very quickly adopted by the artificial insemination companies. It was actually we can discuss that later, they could enter into this technology quite easily for a number of reasons. And remarkably, the predictions that were made in this paper that I showed you before were essentially verified. It was a little bit less than what the simulations were telling us, but it was quite remarkable. So in what people realized, that they had the tools available to identify the bulls that were the top bulls without having to go through this progeny testing. And in no time did the industry adopt this technology. And at the present time, wherever you go in the world, the AI, the bull studs, are using this methodology to select their bulls. Hundreds of thousands of animals are now being genotyped every year for selection purposes. Just mention very, very quickly one thing here. Is that, so these methods are relatively sophisticated. They have very simple forms, and then have more sophisticated forms. The simple form is called GBLUP. Essentially what it does, it fits the infinitesimal animal model. So what people think sometimes when we do genomic selection is that we have, in fact, done a smart GWAS, but that we are capturing information from peaks. They might not be the Manhattan peaks, but they are still peaks. The reality is that, with few exceptions, the signal is actually looks as if it was spread uniformly across the genome, exactly like R.A. Fisher was suggesting. And so that's why I'm saying here, the infinitesimal model, le retour. In fact, what we think is happening at the present time is that rather than doing GWAS, the markers just allow us to reconstruct the genealogies without needs for genealogical records, and most importantly, explaining so-called Mendelian sampling. If we look at a crop of full SIPs, they have exactly the same pedigree, but they don't inherit all the same genes from their parents. This is what genomic selection using GBLUP is actually capturing. And so the impression is that the infinitesimal model is actually coming back. So I think this has, so this is nearly my last point, I think this has bearing in human genetics. So you've all heard of this missing heritability thing. So GWAS studies have been conducted with enormous success in human genetics and have identified and are identifying a growing number of loci influencing predisposition to common complex diseases. But when you do a bit of the math, you realize that you only explain a small fraction of the genetic variants, the so-called missing heritability. Now, a number of people, including especially Peter Vischer, have hypothesized that what is observed in these cows might actually apply as well to these common complex diseases. And although height is not a disease, he has applied exactly the same methods as the one that are used in genomic selection to try to capture more of the genetic variants in terms of height. And this worked, in fact, remarkably well. So a number of people are now exploring exactly the same approaches, not for a trait like height, but for a series of other diseases, or for diseases. And my conviction is, in fact, that we have a good chance to come up with the same kind of results as the one found in animals. So now we are at the plateau of productivity when it comes to genomic selection. So this means that people are trying to fine-tune the methods, expanding them to other breeds and to other species. But I'm quite convinced that with what we have seen in dairy cattle, the chances of success are extremely high. So what will happen in the future, and this is my last slide, I apologize. So essentially what I think will happen is that, fortunately, we will go back to function. So to some extent, genomic selection has been not only the return of the infinitesimal model, but with that, the return of the black box. So this whole thing seems to be working, but essentially without any knowledge of the genes that are being affected. And for a number of reasons which we don't have time to discuss, it's the conviction of several people, including me, that going deeper into what is really happening will be essential to move the barrier, to move the limits of this technology up. And there are efforts towards that way. The first is that we will increase the marker density. Agricultural geneticists are pusillanimous, we say in French. They always try to not spend too much money. I think it's time to do like in human genetics and dare to do things with very large cohorts and many markers. And the sequencing, the advances in sequencing now will probably allow us to increase the density very, very high. This will actually cause a problem of overfitting, which will require to be able to put better priors on specific SNPs for which we have reasons to believe that they are the causative SNPs. And this will require the development of projects that are akin to ENCODE projects applied to livestock. And so it's my impression that this is the direction in which genomics in livestock at the present time is going. So I'll conclude just by saying that I think that we have witnessed in livestock genomics, and especially in dairy cattle genetics, the kind of success that is a once-in-a-lifetime story. See that in a few months, something that was a concept has really been translated into application adopted by a very large fraction, if not the entire industry, in dairy cattle. And two, the fact that the methodology relies on a principle, so the fact that for complex traits you have a very large number, a quasi-infinitesimal model, a concept that works in this context, and this, I think, bears some importance for equivalent or traits with equivalent architecture in human genetics. And I thank you for your attention.